Alright, in this lesson we are going to cover accuracy versus precision, as well as the scientific method. So if you want to go ahead and write these as the label on the top of your notes. First thing we need to do is define accuracy and precision. So we'll start with precision. And precision reflects the reproducibility of a given type of measurement. So that means how many times you can reproduce the same results, or how many times you can get the same answer. So to remember precision, think of it as, did you get the same answer each time? So if you took some results, or you're gathering results, and you got the same answer each time, you could say that your results are precise. Accuracy reflects how close a group of measurements are to an accepted or the correct value. So accuracy is going to look at, did you get the right answer or the correct answer? Okay, so those are the definitions of accuracy and precision. Now I want you to go ahead and draw 3 dartboards, and I want you to put the darts kind of how I've done it. If you want to just draw quick dartboards and then put dots for the darts, that's fine. So we're going to label these as accurate, precise, not accurate, not precise. So looking at our first one, this is number 1. All the darts are very close together, but they are not towards the middle, which is what we want. We want the target is in the very middle. So because they are all close together, it is precise. However, it is not where it's not towards the middle, so it is not accurate. If you look at number 2, all 4 of the darts are in the middle, which is where you want them to go. So therefore it is accurate. They are also all close together, so it is also precise. And then example number 3, the darts are kind of all over the place. So they're not close together, they're not in the same spot, so it is not precise. And they are also not all towards the middle, and so it is not accurate. Okay, so that's an example if you're looking at dartboards. Next example, we're going to look at a data table. So I want you to copy down this data table. Student A, Student B, and Student C, Trial 1, Trial 2, and Trial 3. Let's say this is an example of measuring the length of something, and I'm going to say that the length, or the actual length of it, is going to be 10.2 centimeters. All right, and so that is the desired value or the accepted value, and we'll make up some results for student A. So student A is going to measure that, and trial 1 is going to get 10.0 centimeters. Trial 2 is going to get 10.2 centimeters. And trial 3 is going to get 10.1 centimeters. All right, so if we look at that data, all those numbers are very close together, and so it will be precise. Those numbers are also all close to the accepted or the desired value or the correct value, so it is also accurate. All right, student B is going to get 5.3 centimeters for trial 1, 8.2 centimeters for trial 2, and 10.5 centimeters for trial 3. So even though one of the trials is kind of close to the accepted value, they're not close at all together, and if you averaged them out, it would not be close to 10.2 or our accepted value. So student B numbers are not close to each other, so it is not precise. And is also not accurate because the values do not average close to the accepted value. Student C is going to get 7.1 centimeters for trial 1, 7.1 centimeters for trial 2, and 7.3 centimeters for trial 3. So student C, those values are very close to each other, so it will be precise. However, it is not close to the accepted value, so it is not accurate. So more often you're going to have to look at a data table and look at numbers to determine if it is accurate or precise rather than a dartboard or something like that. So this is an example of data that you might have to look at and classify as either accurate or precise. Okay, now we're going to look at scientific method. This should be semi-review. You should have seen this in several different science classes. So scientific method, there are 6 parts to it. First, or steps, the first step is you need to have a problem. So a question, something you want to research more, but you need to have some sort of problem that you're starting with. Step 2, you need to make observations. And once you have observations about your problem, you're going to then come up with a hypothesis. And a hypothesis is a testable statement. It is usually an if-then statement. So if I do this, then this will happen. All right, so after you form your hypothesis, then you need to do an experiment. So step 4 is an experiment. And we know that an experiment always needs to have a control and variables. So the control in an experiment is the thing that is not changed at all. All right, so the control is not changed during the experiment. The variables do change, and we know we can have two different kinds of variables, independent and dependent. So now make two little subgroups under variables. We have independent and dependent. So the independent variable is something that is changed or measured. I also like to think of it as independent is what I change or what I decide. Or what you determine. Right, so if you're trying to measure fertilizer and how it affects plant growth, something that I could change is how much fertilizer I put into the plants, into the soil. The dependent is what responds to that change. So in the example that I just used, the dependent variable would be how much the plant grows. I can't tell the plant to grow a certain amount, but what I can change is the amount of fertilizer I put in, and what responds to that, the dependent variable, would be the growth of the plant. So step 4 is the experiment. Step 5 is going to be recording the data or performing any calculations. Data is going to be recorded as charts, tables, or graphs. And just if you're going to be graphing it, if we have an X and a Y axis. So this is our Y and X axis. So the dependent variable is always going to be on the y-axis. Okay, so you will always put that label like right here. And that label is going to be your dependent variable, always goes on the y-axis. Your x-axis is going to be your independent variable. And an easy way to remember which one goes to which axis, if you just remember dry mix, so we'll spell this out, D-R-Y-M-I-X. So this stands for dependent is the one that responds and is on the y-axis. So it's the variable that responds. And the dependent is on the y-axis. Mix stands for it's the one that's measured. It is the independent. And it is on the x-axis. So if you just remember dry mix, that'll help you remember which variable goes on which axis when you're graphing them. And then the last step of the scientific method is the conclusion. So it's step 6, conclusion. And so that's where you will state if either your data supports or does not support your hypothesis. And you will write up a conclusion. Also, it is always important to repeat an experiment. For validity, to see how valid your results are. So if you get results one time, you can't quite write a conclusion on it. But if you repeat the experiment and get those same results multiple times, then your conclusion is going to be much more valid. So just to review really quick the parts of the experiment, and you don't have to draw this if you don't want to, but if you are measuring in this picture, it is how much the plant is growing based on what you are watering it with. What you change or what I change is going to be what you are watering it with. So there's Coke, there's juice, and there's water. So that is the independent because that is what I am changing. Independent variable. The growth of the plant depends on what you're watering it with. So that is going to be, the growth is going to be the dependent variable. And we also need some controls, and so the controls are going to be the type of plant and also what the plant is growing out of. So there's two things that are controlled or stayed the same throughout all of those 3 trials.